Coenzyme A is derived from the vitamin pantothenate, and pantothenate is converted to coenzyme A in just 5 simple steps. These steps are universal reactions in all organisms, although they can be catalyzed by distinctly different proteins. The pantothenate kinase catalyzes the first committed step, and it is regulated through feedback regulation by coenzyme A in bacteria and coenzyme A thioesters in mammals. In mammalian systems, feedback inhibition can be reversed by long-chain acylcarnitines and acylethanolamides. There are 2 distinct Pank protein families. Most bacterial Panks have no sequence homology with eukaryotic Panks. The bacterial Panks are regulated by free CoA, whereas the eukaryotic Panks are regulated by acetyl-CoA, or selective Panks are regulated by long-chain acyl-CoAs. Staphylococcus aureus Pank is the exception to both of these points. It has sequence homology with the eukaryotic Panks and does not exhibit any feedback inhibition. Most bacteria, yeast, Drosophila, zebrafish, all of the above have only 1 Pank gene, but mammals have 3 genes and 4 active protein isoforms. Acetyl-CoA sits at the crossroads of intermediary metabolism. It's a key metabolic substrate in both synthetic and degradative reactions. It's also an allosteric regulator of pyruvate dehydrogenase and an allosteric activator of pyruvate decarboxylase. Multiple copies of free CoA are necessary to break down and oxidize fatty acids. Now, CoA synthesis from pantothenate provides a direct pipeline into the control center of metabolism, and thus it is heavily regulated. There is a complex regulatory network to control and maintain cellular CoA levels. As I've mentioned, there's 3 genes and 4 Pank proteins. There are at least 2 Nudix hydrolases that degrade CoA. And the Pank isoforms differ in their responses to feedback inhibition by acetyl-CoA and differ in their responses to feedback activation by long-chain acylcarnitine. The different Pank isoforms also have different subcellular locations. Pank 1 alpha is found in the nucleus, 1 beta associates with endocytic vesicles, Pank 2 associates with the mitochondria, that is the human form. However, the mouse Pank 2 is found in the cytosol, as is the Pank 3 in both species. Different tissues express different combinations of Pank isoforms, which ultimately determine the tissue CoA levels. The tissue CoA levels span more than a 10-fold range, and those with the highest oxidation rates have the highest CoA, such as liver, heart, and brown adipose tissue. In these tissues, Pank 1 is the dominant isoform, and it also is the least sensitive to feedback inhibition. So the Pank expression profile determines, at least in part, the function of the different mammalian tissues. In summary, I'd like to say that CoA is essential, CoA is dynamic, and Pank is a key regulator of CoA.